Good afternoon everyone. Now as I was discussing in the previous topic that dominant and recessive characters, and pea plant has the 7 contrasting characters. That's why Mendel has selected pea plant for his experimentation. Dominant means the characters which are able to express themselves in the next generation, and recessive are those which are not able to express themselves. They remain suppressed in the next generation and not able to express. These are called recessive characters. Now we will talk about one by one. The first character which he took it and which he found that it is there in the pea plant is height. Tall as dominant height and recessive height is dwarf, or we can say that short-heighted. The second character is flower position. There are 2 ways in which the flower is positioned in a plant. The flower position is either axial one or terminal one. As you can see in the diagram, axial means which is born on the branch. That is called axial position of a flower, and the another recessive one is the terminal position of the flower in which the branch ends with a flower on the top. This is called recessive terminal position in pea plant. Third one is flower color. The color of the flower. These are either the purple ones are the dominant ones and the white color flowers are recessive in nature. That means they are not appearing in the next generation frequently. But how are they appearing in the next generation? That we will explain you when we are going to learn about the Mendel crosses, various monohybrid, dihybrid crosses. Next is pod shape. That means the shape of the pea pod. That is inflated is dominant one and recessive is constricted. You must have observed that in pea plant, if you have seen the pod, either it is in this flat shape or it is in the form of constriction, means the pod is in this shape. So that is recessive shape. Another one is pod color. Pod color means either it can be the color of the pod, chilke ka rang, that can be green or it can be yellow. The next character is seed shape. Seed shape, the seed can be round or the seed can be wrinkled, which is having a lot of constriction, less amount of the water. That's the shape of the seed at certain cases, and that comes under the recessive character. Last character is seed color. This is a little confusing because the children, when the question is asked, generally the question is asked between seed color and the pod color. In pod color, green is the dominating character, but in seed color, yellow is dominating and green is recessive. So the main concern for the various other characters, the contrasting traits are absolutely clear. The features are absolutely clear, but these are 2 little confusing, pod color or seed color. So in pod color, if we can learn only one, we'll be able to relate that the other one is opposite of this, so we'll be able to learn better. So pod color is green and yellow. This we can keep in our mind. Next part to this is that we are going to learn about the various important terms that we are learning in genetics. Important terms. Kindly make your notes side by side so that it will be easy for you. Keep copying these things in your notebooks as well. Important terms are, number 1, genes. What are genes? Actually, where they are located. Genes are located on the chromosomes, and genes are the functional unit of inheritance. Functional unit of inheritance, which Mendel has said and name given by Mendel as factors. According to Mendel, these were factors. Next term that we will learn, and this is given by, Johansson. This term is given by Johansson. Now, the next part is, originally it was called factors. Now, the next term that we are learning, alleles. So, what are alleles? The contrasting characters are called allele. A character is height. In height, we have two contrast features. Number one is called tall and the other is called dwarf. So, these are contrasting characters, or we call them as alleles. So, what do we write for allele? That forms of genes. Forms of genes, or we can say that contrasting characters. Character. Fine. Next term we learn, that is dominant and recessive. Dominant, which express itself, is called dominant gene, and next is recessive, which suppress itself, is called, suppresses itself in the next generation, that is called recessive gene. The next one is homozygous. Homozygous. As we all know, homo means same. So homozygous, I'll explain you with the help of, say, we have two genes T, T, and both the genes are similar. So that means they are homozygous in nature. They can be small t and small t. Again, both are similar. So we can say that they are also homozygous. But now dominant genes we are expressing with the help of capital letters like this. And recessive genes we are expressing with the help of small letters. But one thing we have to keep it in our mind when we are labeling these genes, that we are using only single letter for one character. Say suppose if we are taking a character as an example for height. So for height we have two contrasting character features. The one is tall and the other is dwarf. So for tall, if we are taking capital T, so then on the other hand for dwarf we are not taking D, we are going to take it small t. Right? So this is height as an example. Next, homozygous. So this is a homozygous tall and this is homozygous dwarf. Another one, next is that we have phenotype and genotype. Phenotype and genotype. Before we are going to do phenotype and genotype, I'll also tell you that there is another term that we have to know. This is called heterozygous condition. So what do you mean by heterozygous? Heterozygous means in which both the genes are different. That means one is capital, which is dominating in nature. The other one is recessive and small size. So these are hetero. Hetero means different. So this is called heterozygous condition. Now after this, homo means same, either both capital or both small, and hetero means that are different, and one is capital and one is small. The next one is phenotype and genotype. As the name indicates, phenotype means physical appearance. So, how physically it looks like, whether it's a tall or it's a short or dwarfing condition, that is called phenotype. And genotype means genetic makeup. Now with an example I'll explain you. Say suppose if I'm having this as a genes set, then according to this what I will get? I'll say this is tall in nature. What about this one? If I'm going to talk about this is also tall because out of the two genes, if one is capital and the other is small, that means the one is able to express itself and the other suppresses its effect. So that means this is also called tall. But under tall, this is called as phenotype. But genetically, if we are going to see that this has two different genes, one capital and one small, which is heterozygous in nature, which shows that genetically the genotype is different, which I'll explain you with the help of a cross. Say for example, if I'm taking this as one gene set, say for example, for a male gene set, and on the other hand, I'm taking that female with having this gene set, then what is happening here? Under such circumstances, this and this is going to be multiplied together and we will get this as a result. Same way this and this results as T. Another one similar. This is also similar. Now, if I'm going to talk about the phenotype, physical appearance. Physical appearance, I said, if one is capital and the other is small, that means this is taken as the dominating character. So, dominance is tall. So, we say that according to phenotype, all are having same type, we call it as tall. But when we are going to talk about genotype, under such circumstances, what is happening? That here the genotype is different. That is with this as a character. Now I will take another example in which I will explain you that they are different. Say in place of this, I will take two genes, one is capital and the other is small. Same way here, one is capital and the other is small. And I'm going to cross them, I'll get a result like this. Now here, the ratios will be different. Phenotypic ratio, if I'll see all, then under such circumstances, what I am able to get, that this is tall, this is also tall, this is tall, but what about this one? This is short, dwarf in nature. So if I am going to talk about phenotypic ratio, phenotype ratio, so how many tall are there? 1, 2, 3. 3 tall and 1 dwarf. So the ratio becomes 3:1. Similarly, if I am going to talk about genotype, in genotype the ratio is different. It is 1 pure tall. Because I have to see the genetic makeup in genotype. Genetically, how is this? This is T. That is, both capital T's are there. 1 out of 4. 2 are with heterozygous nature and 1 dwarf. So the genotypic ratio is 1:2:1, which is different. And this we have to learn as a term. The phenotype means the physical appearance, which says this as tall. Similarly, this also counted as tall. But when it comes to this condition, it is called dwarf. So the phenotypic ratio and the genotypic ratio is different. Next comes cross. What do you mean by cross? Okay, from here. Cross. Cross means when we are breeding two plants. Breeding two plants for reproduction to happen between them. That is called a cross. And we have three types of crosses. Number one is called monohybrid cross. That means in which we are only taking single character and then we assess. That is called monohybrid cross. Say suppose if I am taking only character as height, so I have taken this as an example. This is called monohybrid cross with one character to be assessed. Next, dihybrid cross. Dihybrid cross means in which di means two. So we are taking two characters. Number one, say suppose tall, and the another character which I am taking here, say the flower color. So the flower color is purple in nature. So I have taken two characters together for one cross. This is called dihybrid cross. On the other hand, it will be like this. The same letter we have taken up, but in the dominating side we are writing these as capital and on the other hand we are writing small. Another one is trihybrid cross. When we are taking three characters all together to explain a trihybrid cross. Say example, we can take up three all together. Tall, purple, as well as we can take up the color. So then seed color. So seed color we can say that capital C, and here we will take up like small c, small c. So this is trihybrid cross. Next to this is generation, filial generation. What do you mean by filial? Next term comes filial. Yes. What do you mean by filial? That means representing generations. Representing generations. Say suppose if I am going to write F1, this means this is the first generation for which we are talking about. F2 means next generation and F3 means another next generation. So we are representing various generations in genetics. While doing our process, then we are using this word as filial generation. F1, F2, F3. Next comes Punnett square. Punnett square is actually a mathematical representation or a pictorial representation of the various genes. How the genes are segregated and then appearing in the next generation with the help of a table when we are explaining. That is called a Punnett square. Say for example, if I am having this, and I am going to write and then explaining them, this results into homozygous tall, this results into heterozygous tall, again heterozygous tall, and this is short. So then this representation, this type of the representation of the various genes that under such table, that is called Punnett square. Next is called selfing. These terms are important because when we are learning ahead, then these terms we are going to use in the explanation. Next is called selfing. Selfing means self-test. That means to come up with pure line, we require selfing. Same genes crossed with another similar type, that is called selfing. Next one is called pure line. To get the similar type of the characters generation after generation, when we are crossing them similarly again and again, that is called pure line. To get a pure line character, Mendel has crossed the same characters again and again for at least 5 generations, and that is called pure line. A particular genotype crossed for minimum 5 generations is called pure line. Genotype, when crossed again and again in various durations, and minimum for 5 generations, that makes the pure line breed. Pure line. Thank you.